Antibiotics have been a cornerstone of modern medicine since the discovery of penicillin in 1928, revolutionizing our ability to combat bacterial infections. These powerful drugs work by targeting specific features of bacterial cells, disrupting their ability to grow, reproduce, or survive. Unlike human cells, bacteria have unique structures and processes that antibiotics exploit, making them effective weapons in the fight against infection. But how exactly do antibiotics work? Let's explore their key mechanisms of action. Inhibiting cell wall synthesis. One of the most common ways antibiotics attack bacteria is by targeting their cell walls, a rigid outer layer that human cells lack. Bacteria rely on this wall for structural integrity and protection. Antibiotics like penicillin and vancomycin interfere with the synthesis of peptidoglycan, a critical component of the bacterial cell wall. By blocking the enzymes responsible for building or cross-linking peptidoglycan, these drugs weaken the wall, causing the bacteria to burst under their own internal pressure. This mechanism is particularly effective against gram-positive bacteria, which have thick peptidoglycan layers. Disrupting protein synthesis. Proteins are the workhorses of life, and bacteria need them to function and multiply. Many antibiotics zero in on the bacterial ribosome, the molecular machine that translates genetic code into proteins. Drugs like tetracyclines, macrolides, for example, erythromycin, and aminoglycosides, for example, streptomycin, bind to different parts of the ribosome, halting protein production. Without essential proteins, bacteria can't carry out vital processes or reproduce, effectively stopping the infection in its tracks. Since bacterial ribosomes differ from those in human cells, these antibiotics can selectively target the invaders, blocking DNA replication and transcription. Another strategy antibiotics use is to disrupt the processes that allow bacteria to replicate their DNA or transcribe it into RNA. Fluoroquinolones, such as ciprofloxacin, target enzymes like DNA gyrase and topoisomerase 4, which are essential for unwinding and copying bacterial DNA. Without functional DNA replication, bacteria can't divide. Similarly, rifampin inhibits RNA polymerase, the enzyme responsible for transcribing DNA into RNA, stalling the production of proteins and other molecules bacteria need to survive. These drugs are especially useful against rapidly dividing bacterial populations. Interfering with metabolic pathways. Some antibiotics take a more indirect approach by targeting bacterial metabolism. Sulfonamides and trimethoprim, for example, block the synthesis of folic acid, a vitamin bacteria need to produce nucleotides, the building blocks of DNA and RNA. Unlike humans who obtain folic acid from their diet, bacteria must synthesize it themselves. By mimicking the molecules involved in this pathway, these drugs trick the bacteria into shutting down their own production line, leading to their demise. Damaging cell membranes. Finally, certain antibiotics attack the bacterial cell membrane, a barrier that regulates what enters and exits the cell. Drugs like polymyxins disrupt the membrane's integrity, causing it to leak essential contents like ions and proteins. This loss of control is fatal to the bacteria. While this mechanism is potent, it's often reserved for tough cases, like infections caused by gram-negative bacteria, due to potential toxicity to human cells. A targeted approach with limits. Each class of antibiotics has a specific mechanism, often making them effective against particular types of bacteria, gram-positive, gram-negative, or both. However, their power comes with a catch-overuse and misuse have led to antibiotic resistance, where bacteria evolve ways to evade these mechanisms, such as pumping drugs out of their cells or altering target sites. Understanding how antibiotics work is crucial not only for developing new drugs, but also for using them wisely to preserve their effectiveness. In essence, antibiotics are precision tools in the battle against bacterial infections, exploiting the differences between human and bacterial cells. From dismantling cell walls to halting DNA replication, their diverse mechanisms highlight both the ingenuity of medical science and the ongoing challenge of staying ahead of evolving pathogens.